can tell you really a little bit about what progress we've made since really I started doing research on MECFS in 2010. At that time, Ros and I went to an international meeting in Bonn University in Australia. It was quite an exciting concept because it was a combination of researchers and GPs and affected families run by the Alison Hunter Foundation. Christine Hunter, the mother of Alison Hunter, who died with this illness. And the concept was really exciting. So the first day the researchers talked, and I talked about what we were thinking we were going to do. And then the second day, Ros was among the people who talked much more sort of patient-connected and focused. And while I really enjoyed all the research talks, it did strike me when I at the end of that first day that how much of that research is really going to benefit patients with CFS. And of course, for me, coming from an ME/CFS family, I've got a daughter who's had the illness for 25 years, my wife about 10 years. I was very much steeped in what it means to be an ME/CFS family, as well as the other half of my brain, of course, as a biomedical researcher. So I came back really from that with a sort of a plan of action that what we would do would be very much patient focused, and that we wouldn't lose that sort of grounding, and that's what we tried to do. This is my team for 2015. Just briefly go through them. Margaret Ryan on the left is an Irish woman who works on our Alzheimer's program that I'm also part of, but she's got particular skills that we coaxed into the ME program. Just behind her is an honours student who has worked on cytokine analysis. I'm going to show you data from her work today. In the middle is Tina Edgar, who's been my technician for 35 years. I employed her as an 18-year-old, but I won't tell you exactly how old she is now. She's good at mathematics. Erin Sweetman was an MSc student who's just switched to a PhD, and as for the PhD part of what she's already done, she's going to actually complete the pre-clinical studies for our pilot program. And then Alex Noble at the back had ME as a teenager. She's in moderately good health now, and she's working on the diagnostic test. And unfortunately, up in the inset is Angus Mackay came to Auckland from Auckland to work with me and do a PhD. He's what we call a mature student, somewhat older, but in the last 18 months he's been quite unwell, and so hasn't been able to participate, although he's just easing back into the program now. If I don't go over time and Ros stands up and tells me to stop, I'll just say a little bit about what he's been thinking about, and we're going to go off and look at another area with respect to it. So I'll spend a couple of minutes to talk about that. So, having come back then from this conference at Bonn University and thinking about, where can we contribute that really is patient focused. One of the things I thought was really we want an educated health professional workforce, and I realised the simplest way of doing that would be to clone Ros and just put her around everywhere in the country and we'd solve the problem. But that's a little bit unrealistic, even though we do cloning anyway. And so, I hope that I could make a contribution in that area, and I'll tell you a little about that in a moment. Secondly, although we have clinical guidelines for illness, there's no simple blood test you can go, there's no diagnostic biomarkers where, a GP can just order a blood test and say, yes, you have this illness or you don't. And so that would contribute. And, I felt as an ME/CFS family that would be a good thing for patients to, have access to. And the third thing was just really a better understanding of why patients feel so ill, and so why so many systems in their body are affected. So this was kind of our basic sort of starting point back at the end of 2010. And our paradigm is settled into a very sort of simple one really for the illness. We say, okay, the onset is often follows a period of acute viral infection, like my daughter's case, and Angus, who's the PhD student that's worked with me. But it can be stress of other kind, which could be chemical toxin, it could be a bacteria, it could be some emotional stress, in my view, severe emotional stress which causes cytokine storm maybe in the brain, and then we can go with this illness. So I'm not sort of putting my flag at any one sort of onset or not, just knowing there's a range of potential initiating events which can lead to this illness. One thing I want to emphasise, and you'll see why I do this, I believe it's a chronic illness. I've lived beside my daughter for 25 years, or not, she's living in Auckland now, but and I've seen the illness minute by minute over that 25 years. No one can tell me it's not a chronic illness. My daughter's had it for, my wife's had it for about 10. And these figures down here are ones I think I've taken from a Charles Shepherd booklet, and all our other figures are available. That's kind of when I talk about this, I sort of say about 25% of people unfortunately stay in a acute phase of illness. 70% have improvement but can experience relapses, and then some return to their previous state of well-being, and of course that number, I think, different people will have different views on it. The critical thing which I really have taken from the Anthony's website is that in New Zealand approximately 20 to 25,000 people were affected. Now that's amazing when you think that in Alzheimer's disease, which I work on, there's only 48,000 currently. It's going to rise dramatically as we go up to about 2030, but there's 48,000. So this is an illness which affects half as many people as Alzheimer's. I only wish it got half the amount of research investment as Alzheimer's get, because that would make our efforts a lot easier. But it does. It's a good statistic to tell people, to make them realise this is not a trivial illness that just affects a few people in society. It affects a lot of people, and it's very debilitating on their lives. So that's something that we use. Now, I emphasise that our paradigm says this is a chronic illness, and I was shocked last week to see these headlines in the Telegraph in the UK and also the Guardian, presenting still this alternative view that any CFS is not a chronic disease. I don't know how many of you have seen this. A number of people have, because they contacted me in great anger. And so the headline in the Telegraph was that chronic fatigue syndrome sufferers can overcome symptoms of ME with positive thinking and exercise. Now that statement doesn't bother me too much, because my daughter has had positive thinking for all of her 25 years of her illness, and I'm sure it's benefited her, so I'm not opposed to that. And when I look at the word exercise, it reminds me of chapter 19 of Roz's first book, where she has a few lines about people that are in the acute phase of illness, and is talking about generally the benefit of exercise that we all know. It's all good for all of us. But if you can't do it because of the effects on you, she made a comment about you can wriggle your fingers and wriggle your toes, and I can't show you that, and also your facial muscles. And I've always remembered that as a kind of a way. I think it's a very astute way of reflecting exercise within this illness. So it's the next sentence which really, of course, I was outraged by that. Oxford University has found ME is not actually a chronic illness. I don't know how you define a chronic illness. If someone is affected by an illness for 25 years, is that chronic or not? And so this was a study then from Oxford, and they're a highly respected institution. Professor Sharp published a study in top medical journal in the UK, Lancet, although one of the portfolio of Lancet's journals, Lancet Psychiatry. And he was quoted as saying, a minority, that was in quotation marks, believe that CFS is either caused by a virus or is chronic, and cannot be alleviated. So I just wanted to clear at this stage, I'm one of the minority people, if you believe that. The Guardian, on the other hand, presented this from the other perspective in a fierce rebuttal with the ME Association UK said it would strongly oppose the use of graded exercise therapy and so on. So I downloaded the actual article and read it. In fact, I read it on the plane coming out. I just downloaded it yesterday. And I mean, the article is, you can make some conclusions from it, but it's got nothing about whether this is a chronic disease or not. So I was even more outraged when I read the article. So just sort of, well, put my flag to the post. So one of the worst things about that for me was that Oxford University is, you know, a highly esteemed academic institution. The Lancet, as medical students and health professionals look at the Lancet as a journal where they get all of the things that really are saying the right things. And so those young health professionals and medical students who read this will believe this, and it makes our job much harder, I think, in terms of educating that health. But fortunately, in 2013, the Public Issues Forum of the Department of Theology, of all places, in the University of Otago, ran a forum on public attitudes to ME/CFS, and Heather was involved in the organisation. I was sort of there and thereabouts. And one of the things that I wanted to do was to make sure every medical student got an invitation to this and they knew it was on. And in reality, this was going to be the same sort of show as today. Richie, Roz and I were going to be talking. As it turned out, Roz couldn't travel that day, but Richie got there, so Heather stood up and stepped in for Roz and did a great job. But it was packed out, I think, on the day, and a very effective forum. But the important thing turned out not that many medical students came, but some of the tutors from their third-year program came, and so they approached me afterwards and said, would I give a guest slot in a session which third-year medical students are involved in. So this is healthcare in the community. It's a psychosocial medicine, and it's about unexplained diseases. And I talked with permission from my daughter about her story at the forum, the story of her illness as a young teenager. She was 14 at the time. I also talked about our interaction with the health system as a family and the issues and problems that arose with that. And I emphasised the need for empathy and understanding of this illness. So the tutor said, we just want you to talk about that. We don't want you to tell us too much about, you know, the science of ME or whatever. And I've been trying to kind of get that in and so forth, my subsequent things. But they wanted me to get over that to the students. And I have to say, the first one was very successful. And so I've been asking to be part of it in 2014, 2015. And the students have been incredibly receptive. It's really gratifying. Over my career, I've taught medical students quite a lot, medical molecular biology, which is kind of my area, biomedical science, really. And half of them are just not interested. They don't kind of see this as relevant to their careers coming up in medicine. But this was a completely different response. The students were, it was extremely good to teach them, and we had very good interchange. The most commonly asked question at these sessions is, with some anxiety, students would ask, what would I do if I met an ME/CFS patient? And my response was simply you have to validate that they're ill, that they have an illness, and they deserve your respect, even while you're trying to establish the diagnosis. So ANZME supported this in the sense that the first, in 2014, the New Zealand edition of the Charles Shepherd's UK ME Association booklet was given to every student, and then last year the diploma for clinical practitioners. So at least 2 years, and since medical students tend to partner up, I'm hoping that this will all get scrambled among them. They'll have 2 booklets at least to keep with them with knowledge of the illness. So I think, before this, there was no formal training in this illness through the whole of the medical students training out of the University of Otago. That includes the 2 clinical schools. And a medical student who's the partner of one of my research team on Friday said to me that actually what I did with his class, that's the only thing they've had so far. He did say that most doctors seem to accept there's a biological basis for this illness, but he said only the fringe ones really understand it. I'm not sure I was very happy to hear that last comment. But, okay. So that's kind of where we've been involved in education. So the second thing, what we wanted to contribute was, can we try and develop a diagnostic test with a blood screen based on a diagnostic biomarker? And so I just want to tell you a little bit about what progress we've made in that to date. So the 2 young women that have been involved in this, Erin, who was doing a master's, now switched to a PhD, and Alex is doing a master's. So the starting point is, of course, there's no specific diagnostic lab test, no definitive diagnostic biomarkers, as is quite usual for any disease. You see in the literature, people kind of propose this might be one or that might be one, and then other groups sort of try to use it and find it doesn't work with their cohort. But it's a normal way in which diagnostic biomarker tests develop. It's slow and it's agonising at times, but hopefully eventually one gets to an end point. And as I've said here in the second book, despite Roz's huge effort going around the country educating health practitioners, many still view the diagnosis of any CFS with uncertainty, and I've had interaction with some, and they're very, they lack confidence of being able to do it, despite these important books we have, and Roz obviously was part of the development of this one. So in starting our work on this, we said, well, okay, what do we know that is affected in ME/CFS sufferers? And we know that they have chronic immune dysfunction. So we looked in this area to see if we could find some potential biomarkers. And okay, this is a moderately complex slide, but hopefully there's some words up the top here that you'll be able to recognise. For example, viral infection, stress, bacterial infection, and cytokines, which I'm going to talk a little bit more about. So those things can all impinge down on a protein in the immune system, immune cells, abbreviated here as PKR. It actually stands for protein kinase RNA activated. That's what the PKR means. And over the right here we have this particular protein. If your immune system isn't disturbed, is inactive. This is a cartoon. You can see it's all floppy. It's not doing anything. That's exactly what happens inside our cells. But if it's activated, 2 of those molecules partner up. They come together. And there's a molecular change. And the molecular change here, I've just circled in red. There's a simple phosphate group added to each molecule. So that's a change in molecular state of this protein. And that's just the sort of thing that we were looking for as a possibility, because potentially we can, as I'll show you, we can actually detect those differences. Interestingly enough, this protein is an enzyme which actually adds a phosphate to another protein. It's down this pathway. Don't worry about what it is, but here's another important protein in the immune cell which has got a phosphate group. So we're also looking at that one. So we're looking at PKR and this other protein which changes its molecular state as potential biomarkers. So the strategy then, the concept that we've developed is that can we exploit the change ratio of this what we call phosphorylated, that's with the phosphate, to non-phosphorylated, that's the floppy and active form of PKR in immune cells, to develop a diagnostic blood test. And so this is kind of, in more pictorial terms, how we're going about doing it. So what Erin has done is she, in sheep, has produced a specific antibody which will detect that phosphate residue in the PKR protein when that site is phosphorylated. And that antibody will detect what I call the on state, or the activated state, the chronically activated state, hopefully in the ME patient. In rabbits, she's produced another antibody which detects the off state, in fact, just the protein independent of the phosphate. And so we've got these two specific antibodies, one produced in sheep, one produced in rabbits, which we're kind of using for development of this test. And that's okay. So again, this is a kind of a slide I would show in a scientific talk, and all I want you to realise is that rabbits and sheep, just like us, we're exposed to all sorts of things during our lifetime, and we build antibodies to them. So we have a whole mixture of antibodies against all sorts of things that we've been exposed to. You know, diseases we know about, like measles and mumps and this and that, whatever. And so it's a real sort of mixture, and that means, that makes it difficult if you want to get an antibody to detect something specifically. So what Erin has done is to take that mixture in each of the two cases and go through a very sophisticated purification selection scheme to select out the specific antibodies just against the things that we want to raise. In other words, we're discarding all of the other antibodies made in the other parts of the lifetime of the sheep and the rabbit, and just focusing on the one that we going for, because we've got a much better chance, if we can do this, to actually detect the change in molecular state in the human sample. So here I want to just show you an actual bit of experimental data from Erin's work, and all I want you to look at is the red arrows, basically. So what you see here is an experiment where we're detecting the off state with her selected and highly purified antibody. And what we can see in control, so we've looked at one ME patient and one control here as a proof of principle. So in the control we looked at, we can detect PKR in the off state, and in ME CFS we see the same thing, but then we wouldn't expect all of the molecules to be in the on state, and you might sort of think that this one's a bit lighter than that, and I'm not sure the significance of that, but it might mean something. But the critical thing really is we're looking over here at the on state, because that's only when you've got the immune system activated. I mean, activated if you've got a common cold, very transiently, but chronically we think if you've got ME/CFS. And what you can see in the control there, at the place we expected to see the protein, we saw nothing, but in the one ME/CFS patient, we could see that activated state. So we're pretty excited with that result. It's only one patient and one control, and Erin's going on to look at all of the bloods we have with our Dunedin pilot study now to see whether this actually follows up. So this is the first step, you know, to developing a diagnostic test then based on this difference in molecular state. And then there's quite a lot of work to be done to get it to what this picture is about, which is the kind of test you would need to have if it was going to be in a chem-path laboratory, so a GP could actually ask for it to be done. And this is what we call an ELISA test. And so what we would do, we would have fluorescent markers on our antibodies, and we would mix them together with different fluorescent markers so that they could be detected independently, and then an ELISA could be done on the blood of a potential sufferer, and the idea would be we'd look for that change in phosphorylated status, and you just have to exclude people that obviously had a cold or something, you know, a viral illness where you might have been activating the immune system. Okay. Now there's one other thing I just want to sort of speak about, because Erin did another side part of her master's project, and that was that, okay, we know that multiple things can initiate the illness, like viral infection, stress, bacterial infection, whatever. What is more controversial is what maintains it. Why don't ME patients, like other people when they get Epstein-Barr virus, why don't they just slowly recover and get back to normal within, you know, a few weeks? And is that because they're still carrying the virus, or do they have another virus which might be interacting, or whatever? So that's still very controversial. What we wondered whether, in fact, to maintain the illness, there might be a genetically susceptible subgroup that uses its own molecules to sustain the illness. So what initiates it, say, would be a viral RNA. We might have RNAs which are naturally part of our physiology, which could actually sustain the illness. So what Erin has done is we produce this protein in my lab by genetic engineering so we can get plenty of it. So she did that, and then she isolated RNAs from human cells, and we mixed those together, and the aim was we wanted to see if there were any of our RNAs that would bind to the PKR just like a viral RNA, and then we isolated those and went to New Zealand Genomic Limited because it's a very detailed, quite difficult analysis to analyse, and so we could afford to give them 3 samples, $5,000 for the 3 samples. And the amazing thing, and we actually went for short, tiny RNAs, and I'll talk about these in a minute, but what they found was that there were 14 very small RNAs which seemed to bind to this protein in the same way as the viral RNA is. So this is, again, this is the first step to try and understand the process of what might be maintaining the illness. We might be using 1 or 2 of this in this list to actually keep that protein activated so our immune system is constantly sort of stimulated. So that's kind of where we are on that. Now for the last section I want to talk about stepping back and trying to look at as many molecules in the blood samples of the patients and controls that we've collected and in our studies, and to see if we can get some idea and understanding of what's really going on in the multiple systems that are being affected in the patient. My inspiration for this was a study that was published in the journal Cell in 2012 with the lead author Michael Snyder, who's a geneticist at the Stanford School of Medicine. There were about 70 authors on the paper, and this study cost $2 million, and so some people are quite scathing about it because it's a very self-indulgent study. He took blood samples from himself over a 2-year period and analysed as many molecules as he could, probably, you know, in excess of several 100,000 really, from his blood cells and his blood plasma, to monitor his health over that 2 years. And what I liked about it was this just seems the kind of analysis that we need to be doing for an unexplained disease like ME, CFS. We don't have $2 million to do it for every person, but could we adapt this kind of strategy? And I'll just show you what he found, and it's, you know, in a way it was quite a trivial thing, but so he analysed his own DNA, sequenced his own DNA first, so got his genome sequenced, and then he analysed all of the RNAs, all of the tiny RNAs, all of the metabolites, all of the proteins. And over this 2-year period, he had 2 viral infections. One was a simple common cold, human rhinovirus infection. Another was an upper respiratory virus, nothing that's supposed to be too serious. And what he found was even with a simple common cold, there were about 2,000 of his genes were activated. And about 2,000 were sort of down-regulated, in other words, almost switched off or switched down. Many of these were in things encoding immune functions, and so he was able to determine that. But that is an amazing molecular change just for a simple common cold, and also he detected lots of other molecular changes. What he found through this study when he had the second virus was that he had actually gone into a pre-diabetic state, and there would have been no reason why he would have necessarily had that tested. So spent $2 million to find that he had to have some treatment for pre-diabetes. I don't know if that's good cost-benefit analysis or not. I was quite impressed with the strategy. So we set about to see if we could utilise this kind of concept with ME patients. So the first study then is a Dunedin pilot patient study, 10 patients, 10 controls. The cost for that, again, we've looked to see what we can do that we have to do, but minimally is about $100,000. All of the ME patients, or almost all so far, Ros will see a couple have been diagnosed by Ros. We've got age gender match controls, and so we asked the patients to come in at late morning to give blood at Southern Community Laboratories. They communicated with us as soon as the blood was donated, and my technician here, Tina, raced over and got this blood and started processing it, and we split it into purified plasma, lymphocytes, so one class of blood cells, and neutrophils, and we've used those fractions. And what we're doing now in the middle of the study, we're measuring, we've got the result from the cytokines, I'll show you that, these tiny RNAs in plasma, and then all of the proteins and all of the RNA that are in the immune cells. So that's what we're underway with right now. I can show you a little bit of data which is coming. So in a second study, it turned out that connection through Ros, that Lynette Hodges in Palmerston North is an exercise physiologist, and she was planning a study with ME patients recruited from Palmerston North, and also some multiple sclerosis patients as a control. And I was interested in that because we'd always intended to try and get control patients from another fatigue illness, and as well intends to get normal controls, although that's yet to be collected. So blood samples were collected before exercise, after, and then 24 hours after of a recovery period, and immediately before a re-exercise, and she's measured the exercise parameters. So we've measured the cytokines so far in this, but we haven't raised money to do all of the others in this study, so that's still something we have to do if we can do a complete study on this. So Lisa, for her honours project then, has done the plasma cytokine analysis for these 2 studies. And this is the cheapest part of our analysis. It's cost us $5,000 per study. And we've evaluated changes in 27 specific cytokines that are in plasma. Cytokines are effectors that are up and down regulated as the immune system sort of goes through its phases. And we found there was someone in the Department of Surgery that's doing cytokine analysis is involved in heart research. And so we've managed to actually partner with them so we don't have to do all the development of this analysis, because they'd gone through all of that and they knew all of the tracks. And the idea is that you have a magnetic bead with an antibody on it, and you put your sample through it, and that captures the particular cytokine, and in one little well will be 27 different kinds of this, and then a secondary antibody system with a fluorescent reporter on will bind to it if the cytokine is there and give a flash, give a fluorescent signal. So that's kind of how the study was done, and here's the results of these studies. So for the Dunedin pilot study, remember we had 10 patients, 10 controls. 2 cytokines were upregulated, IL-9 and VEGF, and these are what we call pro-inflammatory cytokines. So they reflect that there's some chronic inflammation in the system for them to be upregulated like that. So that's a pretty good result for only a fairly small sample size of patients and controls. And we also got 2 of the 27 were down-regulated. See here, interleukin-13, down-regulated in the CFS ME patients. And this one is really what we call an anti-inflammatory cytokine. So we're saying in the ME patients, the anti-inflammatory kind of marker, something which is keeping the inflammation in check, goes down. And then there's another one, IP10, shows a similar sort of profile. So 4 of the 27 cytokines that were measured have significant changes in the patient group. And then we did a longitudinal study on one of our patients who was fairly close to the lab, so it was easy to persuade him in a bad relapse to go and have a blood sample taken. And so what we did, we had 3 blood samples from him when he was in relatively good health. This is referred to as healthy here by my student, which is a little bit of an exaggeration. But he was in quite a bad relapse with the second sample. And what you can see are the black bars are all up. So multiple cytokines are elevated during a relapse. And then they come back into kilter once he goes into a more healthy state again. You can see the greys basically match up quite well. So that was quite an interesting observation, that the cytokine profile does actually, is indicative of a relapse. Well, what we don't know, of course, if you're just going to go into a relapse, does it actually show it before you feel it? And could this be used as a management tool, for example, to know just whether you were conducting your life in a way where you were below the threshold for when you might go into a relapse. So it's just one study we've done. And now what about after exercise? Now this was quite interesting here, illustrated with this cytokine. Here's the controls. Remember, these were 3, at the moment, 3 MS patients, and you can see this cytokine shows no change after exercise and no change 24 hours later. Whereas the 8 ME patients, the cytokine level goes up after exercise and stays up 24 hours later. So again, given some indication that at least the molecular change associated with the post-exercise malaise that we know about with respect to this illness. Because the post-exercise, the patients then were going to do more exercise like that, and I haven't seen the data from the net, but you could almost be certain that they're not going to perform well in that exercise regime, which is quite a well-known fact among patients. So if we look at all of the, or a group of the cytokines that were changing, and this is the ME-CFS as a ratio of MS. So if we should be at this point here, if in fact they were moving together. And what you can see that, again, this is before and after again, the black bars after exercise, I showed you one before, but across a few of them, the ME, CFS patients are responding, their cytokines are responding to that exercise regime. Now, I just want to say what we're, doing now. Really, our plan is to kind of get all of this done by Christmas. Optimistically, whether we will, we'll wait and see. So, our holistic personalomics approach, that's measuring as many molecules as possible. So, these tiny RNAs that you find in plasma, we're going to test about 300, greater than 300, 350 of them. There's about 500 that have been identified in plasma, but we have good ways of testing for about 350. And the cost for that, for 10 patients and 10 controls, has been $20,000. We've spent the money, we've got all the stuff, and I'm terrified that we're going to kind of trip, the technician's going to trip up with a sample or something like that, but we hope that we can get this through with just all of the stuff that we've already spent, so we'll have that data. And then with blood cell RNAs, we're talking about maybe 50,000 or even 100,000, including the cellular microRNAs. This is in plasma. We're hoping that studies can also be completed by Christmas, and this is done with New Zealand Genomic Limited. We're negotiating a contract with them at the moment, and we've got about a 5% reduction in price, and I'm hoping to get a bit more, but that's going to cost about $30,000 for that study. Fortunately, once we do our bit, if we can get the RNA out and hand it over to them, it's their responsibility to make it work. So I'll be a little bit less anxious once we've actually handed over the samples. And then the last one is the blood cell proteome, which is to look at all of the proteins in the blood cells and see if we can see changes that are occurring between ME patients and controls. And once we get all of that kind of data, we can do what's called pathway analysis, where we try and link the changed things up into metabolic pathways, and that will give us an indication of what systems aren't operating sort of properly. That's the sort of overall aim. Now, people say to me, haven't you got the results yet? What I've tried to at least communicate is that you've got to do this stepwise in a very careful way, and it takes a lot of time. But I'm quite optimistic that we're now right in the middle of the study, and we're making good progress. And we're certainly spending all of our $100,000 that we've raised. So I just want to say a little bit, one slide about the microRNAs, because I've mentioned those before. So microRNAs are found in plasma, and remarkably, because we never thought RNA was stable, they're quite stable, and they reflect what's going on in your body. So if you're in a healthy state, you'll have a set of microRNAs that indicate that all of your organs and body systems are working well. But a person with an illness may have some microRNAs elevated or reduced, indicating a part of the body is not functioning so well. And we can identify where the RNAs are actually normally functioning. So, for example, if we identify a microRNA that's up in the blood, it might come from the brain. And so we can then link that to things that are happening in the brain. So, as well as this actually can provide diagnostic markers, just like our changed protein state that I mentioned with PKR. Yeah. Can I take 2 more minutes, Roz? Okay. So here's Angus, and he's, as I said, has been ill for about the last 18 months. And characteristic of his illness is he gets these really pressing sort of headaches, and that is very debilitating for him. He calls that neuroinflammation. So while he's been sort of waiting for this to kind of lift and for him to be able to get back to work, he's been doing a little bit of research, and has come in. We now have talks on Friday afternoons. He comes in on Friday afternoon, and he's got a paradigm now that actually the critical controlling thing for ME is focused in the brain, and it's focused around neuroinflammation. And I'll just show you one piece of data which might support this. So we're investigating this, and we're probably going to write an article about this in a couple of months. So there's been an imaging study, and I noticed that the meeting place had a report of, I think, the California imaging study, but this is one out of Riken, just out of Riken Research Institute, just out of Tokyo, which is indicative that the ME/CFS brain is inflamed. And what they do is they inject a molecule. If it binds to a receptor on a brain cell called microglia, then you get a fluorescent signal. And so what you can see here is this is all brain images from ME patients. This is brain images from controls. And they were able to show in several areas of the brain there's enhanced inflammation signals. Now the microglia turned out to be, they sort of sit there on the sideline, not doing anything unless something is happening to the brain, which is a problem. It might be there's a bit of damage to the brain, and microglia are activated and they go to that site. If we are building up, hopefully I'm not, but if we're building up some amyloid plaques for Alzheimer's while I'm speaking, the microglia will be, they have a task of actually going and gobbling that up if they can manage it. So they're a really important group of brain cells. So this is telling us that these microglia are activated. So they're thinking that the brain is in trouble. And so, you know, Angus and I are now thinking about, you know, what studies could we do to actually sort of develop more understanding of this and see the relevance, you know, to the typical ME/CFS patient. After all, back, I think it was in 1992, the World Health Organization referred to ME as a neurological disease, so it really takes it back to its sort of origin. Okay, I'll just. So I'd like to just express my gratitude to Anthony's, first of all, not only for financial support, but for moral, spiritual, physical and emotional support. It's been quite nice in a way, the administrative part isn't been, because I can have frequent interactions with Heather, which is great. And, you know, it's really important for me and what we're doing to be grounded and close to what Anthony, you know, Anthony's kind of view of the illness and whatever. And, you know, I really thoroughly enjoy reading Meeting Place each time. It's just a rich source of useful information. As I said, getting research money through mainstream investment agencies is quite tough against things like obesity, diabetes and heart disease, but we did get a lotteries grant for the Anderson's Trust in Dunedin, a private bequest, which was fantastic for our study, and the university has supported it. So, you know, I mean, this is kind of an exciting journey for me to be part of, particularly as the illness has embraced my family, and I just hope that I can continue to make progress, you know, which will provide some help for the individual patient at some time in the future. Thanks very much.